Hello and welcome. You are listening to a podcast by the Milner Center for Evolution at the University of Bath. I'm Professor Terry King, your host, and today I'm talking to Paula Cover, whose research focuses on understanding how an organism's environment affects the evolution of traits under natural selection. So Paula, you're looking at one of the pillars of our understanding of evolution, natural selection. So talk me through, what is natural selection for somebody who doesn't know?
Well, first thing, you have to have variation on a characteristic or a trait. If you don't have any variation, then selection can do nothing. As I always tell my students, maybe there's an incredible advantage to having 3 eyes, but we just don't have the phenotypic or genotypic variation to respond to that advantage. So selection, first thing, is the fact that the environment you live, the conditions that you live, might favor a certain trait. Whether you respond to that or not, it depends whether you, as I said, genetic variation and phenotypic variation. And if you have plenty, then natural selection will favor the ones that have the trait that increase your fitness, which often we think about as having more babies, but we have to be careful. It's not just more babies, but good babies, healthy babies that also going to give you grandchildren and so forth and so on. And if that's the case, then the genes that are responsible for causing that trait will be increasing in frequency over time, because every single time that you have a whole population, whoever has genes that gives that traits will increase the chances to making more babies. So therefore, over time, you're going to see an increase on individuals with that trait, and therefore an increase in alleles, which is the little bit of DNA that encodes that trait, also increasing on the population.
So what were you studying when you were doing your PhD?
Yes. I started working on plants and pathogens. I did my PhD at Indiana Bloomington, and I met Kurt Lively. Kurt Lively has been working on the Red Queen hypothesis forever, and I thought this is the most fascinating thing ever. And the idea here is that sometimes populations have to be constantly evolving just to stay in the same place when they're trying to fight pathogens. So pathogens are constantly trying to adapt to the host. And of course, the host is always trying to change. But if they're both evolving relatively fast and the pathogen tends to evolve a bit faster because their generation time is smaller and they have a bit more genetic variation, then they're always on top of the host. So the host is constantly evolving, but it's going nowhere because the pathogen has managed to catch up with it. And I thought that was a fascinating idea. And also the Red Queen hypothesis is one of the explanations of why organisms actually have sexual reproduction, which is another very, very big enigma in evolutionary biology. So if you think about it, what I just said, natural selection should always be favoring the best genotype. So you have the best genotype, you've managed to attract 20 mates, and you can have lots of little babies. But every time you make a baby, if you have sexual reproduction, you're kind of destroying that perfect genotype and combining with someone else. So why would you do that if you just had won the lottery and have the best genotype ever? And it's difficult to explain, and we're still trying to explain why organisms do that. There's plenty of organisms that are successful without sexual reproduction. So why is it that we do it? And the Red Queen hypothesis is one potential explanation for that, because it says that you have to create genetic variation to fight the pathogens, and the pathogens very quickly evolve to you. So you can never stay comfortably at your evolved place because you have to constantly be responding because the pathogen is constantly coming after you. So that's the idea of the Red Queen hypothesis, which has been around forever. And it has been, like everything else, demonstrated in bits and pieces. But my dream from ever was to have a population where I knew everything about the genes and I could throw pathogens at it and I could see the Red Queen hypothesis happening in front of my eyes. And that's how I create the MAGIC population, which is one of the things that has the most citations of all the work I have done, which is a population of Arabidopsis, which has a lot of genetic variation and is completely sequenced. And you can use that to do a lot of natural selection experiments and see how populations are responding to selection at the genetic level.
So tell me about your research.
Right. So what is really exciting me at the moment is trying to understand leaf shape. So plants stay in place and they get their food by photosynthesizing. Leaves are the major way in which they can fixate carbon. So if we think about what natural selection is, there should be very strong selection on having the most perfect leaf that you can possibly have, because it's key for your survival. So it's okay to understand that different plant species, because they live in different places, they might have different leaf shapes as being ideal. But why would you have in one population plants with different leaf shapes? That is very puzzling. And you don't see that very much. Most of the time, the variation in leaf shape is between species. But there's this one species that I'm really interested in studying, which is a close relative to Arabidopsis, that have an incredible variation in leaf shape. And a lot of people think that if we understand which leaf shapes are more adapted to certain climates, we could change, for example, the shape of leaves in crops. It's not difficult to change leaf shape genetically, but we don't really understand what is the consequences of these different leaf shapes. So we're trying to use this plant called Curcuma erythema that has spread all the way from the Mediterranean to Iceland and has this leaf shape variation across all the populations. So we're doing crosses with them and trying to find what the genetic basis of it is. And we're also trying to use photographic recordings so we don't contribute to the carbon cost of field work, and also trying to find out if we can do more work with everybody's pictures of plants everywhere, and trying to see, one, if different leaf shapes are more common in different parts of the world, and two, what is it about the leaf shape that makes them better or worse for the plants. This is an ongoing work. But we do know that, contrary to our first hypothesis, it's not environmentally determined. So if you are a plant mother that makes a leaf that is completely divided, then you have babies that also make leaves like that, no matter what environment you grow in. And if you have completely undivided leaf, your babies will do exactly the same thing, no matter what environment you grow. So it's clearly genetically determined. And for some reason, there is no very strong selection, or there's some inability to respond to that selection for some reason, and we see this variation.
So is there any pattern whatsoever? Is there a hypothesis as to what's going on here?
There's some very strong hypotheses. There is even theories repeated on and on and on that more divided leaves are supposed to keep the leaf cooler better than whole leaves. So the expectation is that warmer parts of the world will have more divided leaves, and cooler parts of the world has the complete leaf. Now, nobody has been able to show empirically that that's the case, which is why I'm really interested on it, because most of the variation is between species, and when you look at between species, there's other things playing a role on what leaf shape they have. So this particular species, because it is the same species going all the way from Iceland to Spain, offers a particularly interesting situation in which you can actually test that. It's very cool. People have built leaves of very thin metal material and put into a chamber of a certain temperature with a certain wind and measure all the physical capabilities of leaves. So under those conditions, that's what you would expect. But yet that's not what you see in nature. So we are trying to do now reciprocal transplant experiments and put these plants in different parts of the world and seeing if we can measure directly whether having a certain leaf shape can definitely improve your fitness on hot versus cold climates.
And I suppose the idea is that eventually with this knowledge, you'll be able to apply it to our own crops to make better decisions about what crops we grow, or potentially change leaf shape to be better, or this kind of thing.
That's right. So it's really interesting because a lot of the times when you're trying to solve a crop problem, you tend to think about the very specific problems. So for example, people do research to try to find what genes are turned on when plant is under drought and then try to modify that pathway. But it could be that you could solve this problem at a much basal level, so the plant not even get to the point of experiencing drought by changing the leaf shape. And it would be something that occurs somewhat naturally in most species or genera. So, not that I think it is a problem, but I think it would be much more acceptable for the public to have a change like that than some GM-specific trait.
Yeah, so let's chat about that, because sometimes people get worried about this sort of thing. What's involved in terms of changing a leaf shape?
So in Brassica, which is the family that I work. So brassicas are your cabbages, your broccolis, your cauliflower. And for those of you that don't know, this is all the same species. So Brassica oleracea is the same species, whether you eat the cauliflower, the broccoli, the cabbage, the Brussels sprouts, whatever it is. So you can see that species has a lot of phenotypic variation. And if you want to change the leaf shape, you can cross with some very close ancestral form and change the leaf shape or the flower shape, as you have already seen with all these. So it doesn't require an introduction by bacteria of a gene that only exists on a completely different species. It's just about a process of selection for increasing the dividingness of a leaf or reducing it as you need.
Now, you've got a new paper coming out soon. So what is it that you've been researching? What did you find?
I mentioned that I've been working with Arabidopsis, and Arabidopsis is a great organism because we have sequenced many times. That means we know all the genetic information that Arabidopsis have. More importantly, we have collected Arabidopsis from different parts of the world and sequenced them also, which means we know where on these instructions there is some variation. Now, the best way to actually identify the genetic basis of any trait is to be doing crosses, but it's really time consuming to do a lot of crosses. The other thing you can do, which we do with humans, which we cannot convince to cross as we like, is to do something maybe some of you have done, which is the ancestry DNA, right? So you can send your DNA out and you can identify whether you have variation, and then you can see, okay, everybody that has diabetes have this genetic variant. Everybody that doesn't have diabetes doesn't have that genetic variant. This is called an association analysis. So in plants, you can cross, and you can use a method called QTL analysis, which is when you cross, you try to see which piece of DNA goes with which trait. Now, there has been a debate for a very long time about what is the best way to find the genetic basis of a trait. And what I've done about 10 years ago is I create what we call a magic line. A magic line is a cross between different varieties of Arabidopsis. We put all that together into one population. And we know exactly what the genetic variation for each individual is. So then you can measure whatever trait you want, and you don't have to de novo genotype or sequence every plant. So that has become something that was very difficult to do. It took me like 3 years to do, and then it took like 10 years to do in wheat, and then another 5 years to do in barley. But it's amazing. Nowadays, most crops have come to the conclusion that building a magic line is the way to go. But when you do a magic line, you are using these crosses. And because you are crossing these plants, the pieces of DNA are divided every time you do a cross. But it takes a long time to become a very, very, very small piece of DNA. You would have to break that lots of times, right? So even though you can say, oh, I think there is some genetic information on this bit of DNA that is important to determine when I plant flower, it's still kind of vast amount of information. Now, that other method I mentioned to you, the association method, it's a much more pinpointy. You find like there's a mutation here, and everybody that has that mutation has that trait. So what I've done on this paper is that we show that you can use the magic line that I created to do both, to do QTL and association. And it's great because you can find much more variation that you would do if you use association alone. Because strangely, by keeping track of these genotypes, there's more information than not knowing and treating everybody as if they are random. And doing this by combining both of these approaches, QTL and association, we were able to find a new genetic variant that explains variation in accumulation of metals in plants. So it's more a methodological paper to show that we can actually find the genetic basis of some really complex traits relatively fast using the magic lines.
So what really strikes me about your research is not only are you developing methodologies to find these particular interesting genes, but then there's implications for use to help us in terms of sort of crops and this kind of thing.
That's the hope, yes. And I think people are becoming more and more aware of the importance of not growing a single variety. And I think the magic lines are particularly powerful for this. It's showing that you don't have to, again, genetically modify that one variety that you like to create the trait. You could create a population select for it and has a little bit of genetic variation that would allow you to do more than one thing at once and create a little bit more variation in the field. And, that should keep pests at bay for a little bit, but also it provides an interesting environment for other beasts that should be cohabiting with you. And when you have like a single genotype out there, sometimes it's very difficult to do so.
So what's next for you?
So I have been really interested recently on truly multidisciplinarity. And I have happened to stumble upon a project with my chemical engineer colleague, which is this idea that maybe we can tell when the soil is happy and when plants are happy in the soil. It is clearly a sci-fi project, but there's so many things that have been sci-fi in the world that got done. I think it's just the time for me to get my hands dirty with some project like that. So we are trying to see whether we can get electric signals from an environment that has a happy soil with the right nutrients, or plants that are happy instead of having a pathogen, that we could monitor that and be able to use that in farms to do long distance monitoring of areas for growing crops. So it is totally sci-fi, but we're very, very slowly just starting to see little things. So something that we have got in recently is that actually plants growing with a very low current running in the soil grow better than plants that doesn't have that. And we have just done a very, very small experiment about that. And it feels like such 1970s research, right? But it's true. So we're just trying to see if we can use something to make plants a little bit healthier under very controlled conditions. But if that works, maybe we can move up to something a bit larger scale. We'll see.
So that's really interesting. So do we know why a little bit of electric current is making the plants? It's like tickling the roots? What is it?
We should know, right? I mean, it's a 70s question, isn't it? And again, that's maybe why science is so fascinating. So the old idea was that every cell has a little bit of polarity, isn't it? And that maybe adding a little bit of electricity just make the whole metabolism work a little bit faster. But nowadays, there's some ideas that actually think that the current running in the soil might be changing actually the nutrients in the soil, or more importantly, the big word, the microbiome in the soil. So maybe it's culturing different kinds of organisms in the soil. And those bacteria that are growing in the soil, because it has a little tiny bit of current going on, are plants that are also beneficial for plants. So you're going to have to wait for the next chapter for us to tell you why exactly it is.
Paula, thank you so much for talking with me. This was a podcast by the Milner Center for Evolution at the University of Bath. I'm Turi King, and thank you for listening. If you have any thoughts or comments on this or any other episodes, please contact us through social media. For more information about the Milner Center for Evolution, you can visit our website.